everyone, I'm Shanjita Shanmugasundaram from Zerotocore. Here we are having a series of videos on vaccines. In today's video, we are going to discuss about the third and fourth type of vaccines, that are conjugated and subunit vaccine. If you haven't watched my first two videos, I recommend you to watch those videos first and then to continue here for your easy understanding. For queries, suggestions and doubts, please don't hesitate to contact me in the given below mail ID. And if you're new to our channel, please do subscribe, like and press the bell icon without fail. With no further delay, let's get into today's topic. But before that, we should brush up about few things. So now, what are vaccines and immune response? Vaccines are acquired immunity to a body which will trigger our immune response and activates T cells and B cells that present in our body. And these B cells and T cells would fight against a foreign antigen and finds a solution. B cells will remind the solution, and whenever the same antigen attacks our body again in the future, these cells would react and destroys the foreign pathogen. In our last video, we have discussed about both of these vaccine types. In live attenuated vaccine, the virulent type of pathogen will be taken and manipulated. The manipulated form of virus will be given as vaccine or obviously with certain rules. In inactive vaccine, they follow the same procedure like live attenuated vaccine, but the only difference here is the dead inactivated pathogen will be taken instead of taking a virulent vaccine. And the more fascinating thing about this inactive type of vaccine is the Covaxin, the vaccine of COVID-19 on behalf of Indian science community by Bharat Biotech is followed by this method for protection. Finally, now conjugated vaccine. Conjugated vaccines use polysaccharide as its carrier, and obviously as a base it carries a small part of antigen too. According to our video, the star type object is an antigen and the triangle shape object is our polysaccharide. So in conjugated vaccine, the combination of antigen and polysaccharide enters one's body. Let's see in detail now. The mechanism is, as I already said, the combination of antigen and polysaccharide enters one's body and propagates into endosome. The lysosome that contains hydrolytic enzyme goes and attaches with the endosome. Further, the chopping process occurs. The activity of hydrolytic enzyme is to remove polysaccharide and separate the antigens. The antigen will be shifted from MH to MHC receptor from endosomes. As soon as the antigen attaches with the MHC2 receptor, the T cells will react on it with the help of T cell receptors. The T cell receptors will fight against the infection and produces T memory cells. Now, we should get in doubt then why polysaccharide sugars are used. Sometimes the antigen may not be soluble. If in that case it is very small, it cannot get attached with the receptors and we should target the antigen to get bind. For ensuring that we use conjugated polysaccharides. And we do have advantages and disadvantages in this method too. It is a cost-effective method. It improves immune and memory response. It is a long-lasting protection. It prevents asymptomatic carriages of disease. The pathogens that remain protected by encapsulation are destroyed in this method, so that vaccination is possible against encapsulated bacteria too. And while talking about disadvantages, the preparation time is more complex and complicated. Vaccine may cause mild side effects. The loss of immunogenicity during conjugations occurs. The immunogenic response is restricted to selected antigen only. And yeah, let's jump into our fourth type of vaccine, that is subunit vaccine. It is also most similar like conjugated vaccine. In conjugated vaccine, you use polysaccharides, whereas here we use small pathogen derived components, and the rest mechanisms are more similar. In our next video, let's discuss about DNA vaccines and RNA vaccine. And thank you for watching.